Hi everyone, my name is Jeongbin. Today I'm going to talk about the paper named Screening Social Communication in Spatial Transcriptomics via Collective Optimal Transport. The paper illustrates on social communication, and it is a key to understand the biological heterogeneity, including cell fate decision, cell differentiation, disease processes, and so on. Social communication includes ligand receptor signaling, neuroimmunocancer interaction, and even extends to the organ level interaction, including insulin-mediated pathways or organism level interaction, including behavioral patterns between different organisms. Social communication can be an important clinical marker, including immune checkpoint inhibitors and tissue regeneration context. Regarding the immune checkpoint inhibitors, cancer cells are well known for evading the immune cell's mechanism by expressing a certain social communication apparatus. And when you treat the immune checkpoint inhibitors, that cancer cells' immune evasion mechanism can be blocked so that the immune cells can easily kill the cancer cells, resulting in the cancer treatment. And regarding the tissue regeneration context, I'd like to explain the figures below. There are two tissues. The left one is normal liver, and the right one is the acute liver failure tissue where acetaminophen is treated to induce that disease. As you can see right now, there is a higher spatial overlap between necrosis-related hepatocytes and macrophages so that we can infer higher cell-cell communication occurrence between these cell types in the disease tissue. There are several methods by utilizing the bulk RNA sequencing and single-cell RNA sequencing to understand the dynamics in cell-cell communication. They include the bulk signal R or SKID5 or bulk RNA sequencing and self-NDP, self-chat, cytotalk for single-cell RNA sequencing. These approaches utilize the geometric mean of ligand and receptor gene expression or the linear Hill function-based model as can be seen in the right formula. However, regarding the cell-cell communication, we should understand the spatial proximity between different cells. So now you are looking at the communication between T cells and cancer cells. And as you can see right now, these interactions should be made within close spatial proximity. However, existing bulk or single-cell RNA-seq-based methods don't consider such a spatial proximity. That's the reason why we should utilize the spatial transcriptomics in terms of cell-cell communication. Please note that bulk RNA sequencing is somewhat mixing the cells, and single-cell RNA sequencing is dissociating single cells when acquiring the transcriptomics. However, spatial transcriptomics can preserve the spatial information so that we can analyze the cell-cell communication in reality and real cells. Spatial transcriptomics has recently attracted a great deal of attention. It has been chosen as the method of the year in 2020 by the Nature Methods, and also the method of the year in 2023, the methods for modeling development, and the method of the year in 2024, the spatial proteomics, are also aligned with the recent popularity of the spatial transcriptomics. There are two types of spatial transcriptomic techniques. The first one is the sequencing-based spatial transcriptomics, which is composed of 3 steps: spatially indexed surface step, unbiased analyte capture step, and sequencing step. Please note that the unbiased and analyte capture step can also be subdivided into poly-A sequencing library and lipodepletion library. And the other type of the spatial transcriptomics is imaging-based spatial transcriptomics, where we treated the labeled fluorescent proofs and we acquired imaging iteratively, and then we decoded the low images to acquire the cellular versus subcellular resolution transcriptomics. 5 different spatial transcriptomic techniques were utilized in this paper, 2 of which are sequencing-based methods, and the other 3 of which are imaging-based spatial transcriptomics. Visium employing the in situ printing barcoding and slice burden 2 utilizing the bit-based barcoding are classified as sequencing-based spatial transcriptomics. They utilize the protein tissue or FFP tissue, and they capture the poly-A-tailed RNA when acquiring the transcriptomics. Please note that Visium is somewhat lower in resolution, around 55 micrometer compared to the slice version 2, which is somewhat between 10 micrometer and 2 micrometer. And the star map here, which is a kind of image-based spatial transcriptomics, is utilizing the padlock probe, which is about round-shaped molecular apparatus. And when they are introduced into the cells, they are kind of mechanism of rolling circle amplification happens so that multiple copies of round-shaped RNAs are generated within the cells in a way of in situ sequencing. Please note that this is very similar to the Genome platform, which is a commercialized version of spatial transcriptomics utilizing the padlock probe generated by the 10x Genomics platform. And the other two spatial transcriptomics are Morphish and Zixuan Plus, and they can also utilize the cells in addition to tissue compared to the former three different platforms. And also they utilize oligonucleotide and they acquire multiple images in each iteration, and then they decode the raw images to acquire the cellular or subcellular transcriptomics in a way of combinatorial multiplexing techniques and their own code books. And from now on, I'd like to illustrate on the machine learning techniques to analyze the cell-cell communication within the spatial transcriptomics context. Here, the GeoRo builds a spatial proximity graph to identify interactions through membrane-bound ligand receptor pairs. And the cell phone DB burden tree in the right side restricts interactions to cell clusters in the same microenvironment defined based on spatial information. And also, Axel relates the co-expression of ligand and receptor genes to the spatial diversity of cell types. However, the existing methods don't consider the multi-species interaction. Multiple ligand species can bind to multiple receptor species, resulting in competition. Let's look at the figures below. There are two ligands and one receptor, and one portion of the receptor close to ligand 1 can interact with ligand 1 more preferably compared to ligand 2. And also the other portion of the receptor close to ligand 2 can interact with ligand 2 more preferably compared to the other counterpart. So this is what the competition is really meaning about. In reality, 72% of ligands and 60% of receptors bind to multiple species. So the authors of the paper utilized the optimal transport theorem to analyze the multi-species interaction. Optimal transport is defined as how to distribute resources optimally from source to target. Let's look at the figure below. So the person here is now distributing the send piles from the source and target, and maybe we should consider which kinds of trajectories or paths to follow and which portion of the send pile in the source to move first and which other portion of the source to move last. So that kinds of problems are what optimal transport theorem is really concerning about. It has been studied in economics, physics, astronomy, computer science, etc., and it is now widely utilized in bioinformatics these days. Multi-species interaction requires numerical analysis and deals with gene expression distribution. So these two factors, numerical analysis and distribution, that's the reason why the author of the paper is now using the optimal transport theorem. The optimal transport can be framed as an optimization problem. So now we are finding the optimal transport plan given the cost function C. So let's look at the table in the right upper side. This is the example case built in the economics case. So there are 3 different people in the availability portion and 4 different people in the requirement portion. And the people in the availability can distribute their resources to the people in the requirement section. However, how much amount of the resources from each person in the availability should be each person in the requirement section. It is too hard to estimate, and this is what the optimal transport theorem is really solving about. So please note that this matrix is what we are trying to find, and the row sums and column sums are somewhat fixed constraints. So the formula for the optimal transport can be framed as can be seen here, and this is what the definition of the optimal transport theorem. However, this optimal transport theorem should be modified to be collective optimal transport, or COT, to analyze the multi-species interaction. So here, the COT is defined as an optimal transport for multiple sources and targets, and the dimensionality of the optimal transport plan P can be now regarded as N of L, the number of ligands, and N of R, number of receptors, and N of L squared, which is the dimensionality of the conventional optimal transport for each pair of ligands and receptor. So now the formula is now becoming something like that. And as I mentioned earlier, we are now trying to find the optimal transfer plan given the cost function. However, the constraint in the second line is somewhat becoming inequality rather than equality, as I mentioned earlier here. So actually, when it comes to the multi-species interaction, it's not that easy to find the solution given the equality constraint because there is a kind of inherent noise in the data set. So that's the reason why the author changed the conventional constraints to inequality, and then they are trying to minimize unexplained portion of the resources in availability and requirement section. Please note that here availability corresponds to the Bigant gene expression, and the requirement corresponds to the receptor gene expression. And mu and nu are now correspond to the unexplained or uncoupled masses in ligands and receptors respectively. So then how to solve the collective optimal transport? There are 4 different steps. Step 1, cost matrix is combined with spatial distance matrices and spatial cutoff. Step 2, penalization term is expressed as Shannon entropy net loss. And step 3, Lagrange multiplier is applied. And step 4, numerical approach is introduced to find the solution of the equation in the step 3 by using the updating algorithm approaches. Here, the step 1, the cost matrix is combined with spatial distance matrices and spatial cutoff. So instead of the conventional cost function, now we are considering the spatial distances that can be available in the spatial transcriptomics data. And also instead of full spatial distance matrices, we are now using the spatial cutoff so that the ligand receptor interaction beyond a certain threshold value of the spatial cutoff cannot be considered in this real solution of that problem. Step 2, penalization term is expressed as entropy penalty loss term. So here the F, as I mentioned earlier, is meaning about the penalization term for uncoupled or unexplained part in gene expression in ligands and receptors. And now it can be replaced with gene entropy and L2 loss term. And step 3, Lagrange multiplier is applied to solve the solutions for that problem. So Lagrange multiplier is trying to find the optimal values, including maximum values and minimum values for the objective function f given the constraint g. So the optimal values can be formed in the point where the the gradient f is parallel to the gradient g. Equivalently, gradient f minus lambda times gradient g equals 0. So by using the Lagrangian multiplier, we can acquire a certain equation in the first line here. So as I mentioned earlier, we are trying to find the optimal transport plan P, uncoupled mass in receptor mu, uncoupled mass in the other part in nu. And as I mentioned earlier, there are 2 different constraints. So instead of a single lambda value, now we are considering 2 different lambda values, lambda 1 and lambda 2. And as I mentioned earlier, by using Lagrangian multiplier, we can acquire a single equation in the first line of the part. And I want to show you a magic. So by using that equation, I want to add the same term in the left-hand side and right-hand side, as can be seen here. And instead of f0 of something plus something, we can replace it with f of something. So we can acquire the third line of the equations. And then it's not that easy to find the solution for that equation because the f is so complicated in reality. So we replace with the left-hand side to something similar, something related to f plus 1, and we can replace the right-hand side something related to f else superscript related terms. And then we can consider this type of situation. So now we are considering x of L in the initial point, and then we can find the functional value of y equals to f of x by tracking the line of that graph here. So we can find the functional value, and now we can jump into the next value, x of L plus 1. So by doing multiple iterations of this approach, we can find the value where the y equals to f of x meets y equals to x. So by doing this type of numerical approach, we can find the solution of this equation, which is hard to find the closed solution form, solution. But there are several problems I could find. So there are two different problems. Basically, the first one is convergence, and the other one is uniqueness of the solution. So for example, here we go right side to find the intersection point of the y equals x and y equals f of x. However, we can go left side according to the convexity of the function y equals f of x. In such a case, the convergence matters. And also, if there are multiple intersection points between y equals x and y equals f of x, then maybe the updating algorithm will lead us to something similar to local minima. So the uniqueness of the solution matters as well. However, the algorithm may yield stable and valid solutions regarding the optimal transfer with entropy regularization, strong empirical convergence across various data sets, and consistent and biologically meaningful research. Please note that entropy regularization is popularly utilized in optimal transport theorem, and this can make this numerical approach something preparant to the convergence and uniqueness of the solution. From now on, I'd like to illustrate on the research part of the paper. The first example of the paper is about the vision data of human skin tissue. And here, as you can see, there is the cell type decomposition algorithm where for different basal cell types, two different spinal cell types, and one granular cell types are visualized. And when you look at the shadow time result, the lowest part of the tissue has a highest differentiation activity, while the highest part of the tissue has the lowest differentiation activity, so that I could name the lowest part of the tissue as precursor cells and the highest portion of the cell as terminally differentiated cell. And please note that when considering the distribution of the basal cell types and the differentiation activity spatial distribution, we can think of this type of tissue as the intermediate between epidermis and dermis tissue. And if we go higher of that tissue, then we can find more structural components related to the epidermis. And if we go lower to that tissue, then we can find more structural components related to smooth muscle. And the author focused on the GAS6 and tyrosine as a potential and remarkable ligand receptors by using the collective optimal transport, or COMOT, which is the name of the algorithm. The GAS6 and tyrosine is innate immunity suppressor and may be related to the unconventional T cells, including gamma delta T cells and MAI T cells or tissue resident T cells. Please note that in many other organs, we can find the conventional T cells, including CD4 positive T cells and CD8 positive T cells. However, in the tissues like skins and gastrointestinal tract, we can find a lot of microbiome. We can find a lot of external factors, environmental factors. In such a case, somewhat immune cell population can be very spatial types. So for example, the unconventional T cells are somewhat prevalent in such a tissue. And in such a case, very meticulous immune regulation mechanism is working, something like innate immunity suppressor or innate immunity activator, something like that. And please note that the interaction strength for each spot can be calculated by summing up the relevant entries of the optimal transport plan, as I mentioned earlier. And the GAS6 and tyrosine interaction is strongest between granular cell and granular cell clusters, as can be seen here. And it is also can be acquired by summing up the relevant entries of the optimal transfer plan. And also, the GAS6 and tyrosine interaction is verified experimentally by using immunostaining and RNA scope. For example, here, tyrosine colored in green and GAS6 colored in red are somewhat coherent spatially, resulting in yellowish mixed pattern. So they are spatially located in the same position. And then the author now focused on the pathway activity by using the COMOT algorithm. Please note that this arrow can be also calculated by computing the vector sum of the relevant entries of the optimal transfer plan. And here we could find the wind pathway and TJ beta superfamily are consistent regarding the direction and the length of the vectors. And please note that wind promotes basal stem cell proliferation while TJ beta suppresses basal stem cell proliferation. When considering the signal distribution, preferably on the lower side of the tissue, which is exact position of the basal cell types, actually it can be an evidence of the basal stem cell related ligand receptor interactions existence within the tissue. Even if wind pathway and TJ beta act antagonistically to each other, it can also be a great indicator of meticulous regulatory mechanisms to mediate the basal stem cell proliferation. And also we can find the genes associated with a certain pathway regarding the ligand and receptor interaction. So as I mentioned earlier, we can find the signal strength for each pathway by summing up the relevant entries of the optimal transfer plan. So for example, regarding the wind, we can order the spot with the lowest ligand receptor interaction strength to the highest spot. And then we can compare this type of ordering signal with the gene expression gene by gene. And then as a result, we can find keratin 15 and keratin 5 are positively associated with the increasing order of the received signals in wind pathway and TGF beta superfamily. And also loricrin and filaggrin are having somewhat negatively association with the increasing order of wind pathway in TGF beta superfamily. Please note that keratin 15 and keratin 5 are basal markers, and loricrin and filaggrin are granular markers known for the negative regulator for basal proliferation. So that's the evidence of the implication of basal proliferation mediated pathway by using ligand receptor interactions. So as I mentioned earlier, Wnt is related to the basal stem cell proliferation, and the similar result can come up with this type of gene association studies as well. And also this algorithm commode can be applied to mouse brain barfish data. So here you can see the cell type decomposition results where each cell is expressed as different colors, and this same color coding applied to the networks between different cells. Please note that the arrows indicate the different ligand receptor cell-cell communication, and each box represents its different slices of the tissue. And for example, excitatory neurons appear multiple times across different slices of the tissue. However, certain cell types like parasite-related cell-cell communication only appears only some part of the tissues. So it can reveal the biological consistency and also heterogeneity at the same time. Also, this analysis can implement spot embedding. As I mentioned earlier, the spot can be represented as a high-dimensional vector, which has a dimensionality of 2 times number of ligands times number of receptors. By using the high-dimensional data, similar to gene expression data, we can also cluster something like here. And then maybe the result is partially consistent with the cell type result. So it is also showing biological reproducibility and biological heterogeneity at the same time. From now on, I'd like to discuss the conclusions, starting with advantages. So this algorithm accounts for collective or global aspects of cell-cell communication, including competition between ligands, receptors, and even cells. And this algorithm enables a variety of downstream analysis, including signaling direction, cluster-level communication, signaling dependence in expression, etc. And commode analysis has been applied across 5 different spatial transcriptomics platforms and validated in diverse biological systems, including skin tissue, brain tissue. And this algorithm is implemented in Python and easy to learn. So I learned that algorithm by myself a few days ago. And what we need to do to prepare the data is just library some normalization and log transformation. And then we should learn just 24 of the lines of the code, as can be seen here. So that algorithm is so easy to learn. However, I'd like to mention several limitations to note. For example, this algorithm utilizes the 1000 micrometer cutoff for cell-cell communication, and it may seem biologically unrealistic because the typical cell-cell communication occurs within 10 to 100 micrometer. And also, this type of parameter should be tuned to different spatial transcriptomics platforms and different biological systems. And also, as I mentioned earlier, there is no mathematical consideration for the convergence or uniqueness of the solution. And the current method does not consider the protein abundance and protein modification or something similar, protein information, resulting in unreliable false positive ligand and receptor pairs. For example, if a certain ligand receptor pair come up with this type of analysis, if such proteins are so quickly degraded, then we cannot easily be convinced with the biological existence of that interaction within the tissue. So we should understand the protein information as well regarding the cell communication. However, there are several solutions for that. For example, here, as you can see in the below figure, there are several spatial multiple profiling or mass spectrometry based spatial proteomics platforms can also be aligned with this type of analysis to support the ligand receptor pairs acquired from this algorithm. For example, this spatial multiple profiling is provided by 10x Genomics very recently, and it can provide dozens of protein expression data. And also the problem I want to mention is the triple interaction. Triple interaction cannot be analyzed by using commode analysis because it is only considering the two-party system regarding the cell-cell communication. Now you are looking at the triple interaction of XCR2A as a ligand, ITGAX as a receptor, and ITGB2 as a co-receptor generated by alpha-4-3. Okay, this is my last of the slide, and I'd like to appreciate to Kim Fai, Jeremy, Jiwin, and Abilab. Thank you. And any question? Yeah, Jiwin. Yeah, so that's a very important question. So as I mentioned earlier, the commode analysis relies on the spatial cutoff, and there are several parameters, but it is not that easily to be determined in reality. So maybe, for example, different spatial transcripting platform which has different spatial resolution cannot be that optimal with the default setting of the commode analysis, but it's not easy to find the optimal parameters for each different tissue. So maybe it should be compromised in reality. Yeah. Here, yeah. Yeah.
So it seemed like to me, like my understanding was that they kind of tailored the interactions between ligands and receptors based on competitive binding, but there are other types of like inhibitions binding patterns. So how do you think they could include that more like realistic perspective of ligand receptor interaction?
Yeah, that's very important. Yeah, question. Yeah, that's very important question. So you mentioned that, so there are several complicated interaction mechanisms like competitive inhibitors or competitive inhibitors, even non-competitive inhibitors. There are a lot of variety of interaction between different proteins. So as I mentioned earlier, the only solution I could think of is some protein information. For example, AlphaFold can also be the tool to make that analysis result more accurate. So I think I'm believing that when we introduce the proteomics models, AI model into the ligand and receptor analysis, then that could be more accurate just because of their region as well. Yeah. Ingrid? So as far as I know, the entropy has nothing to do with spatial diffusion of the signal, but only related to the loss function. So you can also use mean square error or error loss. So a lot of variety can also be possible. So I think I do believe that if we introduce the spatial diffusion, it also makes sense. Yeah.
So the paper mentions, like in the discussion portion, the emergence of temporal sequences. How do you think they could expand this model to handle some of that temporal data for the cyclic signaling or some of the feedback time-based interactions?
So yeah, that's very important. So when it comes to the interaction dynamic matter, and I could come up with two different solutions. So as I mentioned earlier, the optimal transport is somewhat similar to distribution from sources to targets. And when I think we can maybe design the cost function related to times. This is my first strategy. And the second strategy I could come up with the AI proteomics model beyond the AlphaFold 3. I think there are a number of approaches to illustrate on dynamics between ligands and receptors. For example, a certain interaction would be very conservative across times, while the other possible interaction can be very changing quickly. In such a case, we can introduce that information to the ligand receptor interaction versus a communication context. Yeah, that's my answer. Yeah. Yeah, that's right, in burst. Yeah, that's a very important point. So as I mentioned earlier, Wnt pathway and TGF-beta superfamily have similar spatial pattern. That's very good, important. That's very great significance regarding the basal stem cell proliferation. However, the problem is they act in reverse direction. Actually, it's hard to answer right now. So actually, bioinformatics is something problematic because of that reason. We could come up with various types of results, but it's hard to understand ahead in some times. So to understand the mechanism more accurately and more explicitly, maybe we should introduce perturbation studies. But that's my conclusion in general cases. Yeah, that's right. So, for example, knockout or overexpression of that certain genes can also be very important to identify which one is first and which other one is left or at the same time or not. So that could be the possible one approach, and also we can also inhibit that gene expression by treating the inhibitors, or there are a number of approaches to do that. It's generally termed as perturbation study.